enzyme, GTP molecules will be converted to cyclic GMP. Guanine instead of adenine as the amino acid. Very similar in effect. This is now our second messenger. And downstream, it will activate additional protein kinases, which will phosphorylate or dephosphorylate other molecules. What enzyme degraded cyclic AMP? Phosphodiesterase. Guess what enzyme degrades cyclic GMP? Same one. So that's we don't have to memorize another word. So this will also degrade cyclic GMP. The advantage? Rapid production of a second messenger. Disadvantage? Loss of what? Control. Loss of regulation. Increased chances that cyclic GMP might be produced when we don't want it to be produced. So first example of a pathway for a membrane-bound enzyme, a membrane-bound receptor that doesn't utilize a G protein. And the next pathway, we're going to do it one way. I don't have eyes on the back of my head. The next pathway, we're going to leave out, I guess I'll just erase this one. We're going to leave out even all of this, and we're going to go directly to the protein kinase step.
You're not erasing. You're not erasing.
I'll move over to this one.
What's the advantage again of the, you said
Rapid
We
production.
still have the amplification, not as much of the amplification, but a more rapid response. Because we're not doing the G protein and then the enzyme activation, so we cut out a couple of steps.
Yeah, rapid production of second messenger.
Now we're going to remove even that small step. So in this particular pathway, we have absent the G protein. We have absent the membrane-bound enzyme. All we have is the receptor. Hormone binds to the receptor. The receptor itself is still not an active channel, so we have to have something happen inside the cell directed by something else, at least the, but the receptor with the hormone becomes the enzyme. Phosphate ions will attach to it, and now mu proteins, not mu proteins, but unphosphorylated proteins will interact directly with the receptor and will become phosphorylated. The receptor becomes the phosphorylating molecule. What is it functioning as? What do we call the other molecules that were enzymes that function to phosphorylate? Protein kinase. So in this case, it is the receptor that is acting as a protein kinase. So you can see that we've simplified and cut out even more steps. So obviously, lecture notes out, and draw the pathways. So get your brain to form those synapses again. Draw them out on a whiteboard or a piece of paper and say, okay, what was this pathway? What's the pathway where the receptor takes care of things? What's the pathway where I have that really long series of words? And write them out, looking back to see how to spell those words. But until you can understand it, okay? You can differentiate between them. Don't stop there though, okay? Obviously not in the same hour or 5 hours. Take a break, take a day or so. And then come back, and this is where the use of flashcards, not just for pure memorization, but to allow you to more quickly associate ideas or look at comparisons and contrasts. Obviously, you could just write them down on a piece of paper, okay? But by the flashcards, you can just quickly review what they do too. But don't just use them for that. That's the linear learning, all right? Isolating each pathway and the steps that occur. Now try to blend that information and ask yourself a series of questions, all right? Can you name, of all the molecules we talked about today, what molecules are membrane bound? So if you had your flashcards in hand, you could kind of fly through them and at each one make that decision. Right now you're going to have to either look at your notes or stick it up in your head. Okay, so of all the molecules we talked about in those 5 different mechanisms for activating, utilizing a hormone receptor, which of those molecules were in the membrane?
Receptor?
Obviously, good. A lot of people forget that one. So the receptor is in the membrane.
GDP.
The G protein is in the membrane.
Calcium channel?
Our calcium channel is in the membrane, at least the plasma membrane one, not the RER one.
And cyclases?
Cyclase ATP? No.
Adenylate cyclase?
ATP.
Adenylate cyclase?
Adenylate cyclase are many of our enzymes first. So adenylate cyclase.
Phospholipase C.
So what you really mean are which ones are integral? Or which
Membrane
ones are
bound.
actually?
Pardon? Are in the
Integral or
integral part of the
peripheral?
membrane. So that's where they,
Well, that's what I meant.
that's where we would find them. That's what you meant for what?
When I said cyclases.
Oh, when you said, I was thinking cyclics, the cyclic
Oh, oh.
GMP, sorry. Phospholipase C. I'm sorry, I was not hearing the cyclase part of that. One like cyclase. Okay.
Phosphoinositol.
Our lipid, phosphoinositol, thank you for not using the abbreviation.
You know, the book gives a different name. Which one do you prefer?
Either one, if it's in print and you're not the one that printed it. Just like adenylate cyclase or adenylyl cyclase. You can show it to me somewhere.
So if it breaks down in the membrane also just to the
Yeah.
other inositol triphosphate and
That one's not
Cyclase.
membrane bound. But the other one, the diacylglycerol is.
Glycerol is. So this one's membrane bound, not membrane bound.
All right, so stopping here, all right, obviously you could look those all up. But that's why I want you to wait until you study these pathways. And I don't want you to write this down and memorize it as a list. That would be a little difficult to try to recall. It'd be, and you're making your brain work if you study the pathways first, can visualize them, can draw them out, and then from that knowledge base, not the list of information that's in your lecture notes and in your textbook that you're just copying onto the piece of paper, recall it, visualize it happening at, yes, that is in the membrane, because in my mind's eye, memory bank, I can see that those activities in that particular molecule never left the membrane. So you can almost feel your brain getting tired while you're thinking of while you're doing this work, okay? So, other questions? You can come up with all sorts of questions. What molecules function as a phosphorylate, add phosphate groups?
Protein.
Wait, can phosphorylate?
Okay, yeah. What molecule
CM? Okay. Camp?
are phosphorylators?
Camp?
No.
Oh,
What does CAMP activate? CAMP
Oh,
is
the
cyclic
G12.
AMP.
oh, okay, okay.
GDP?
No.
Oh,
Cyclic AMP functions to activate
Protein kinase A.
protein kinase A.
Way to go, Chad.
All right. And diacylglycerols in calcium function to activate protein kinase C. What other molecule phosphorylates?
The receptor?
the receptor.
The receptor itself. Excellent. So then you could take this question and you could turn it around. You could say, what are the molecules that activate phosphorylators? And we would have cyclic AMP and cyclic GMP and diacylglycerol.
Okay.
Okay? So in asking those types of questions now, you're forcing yourself to go back in and dig into that pathway. Do I really understand that pathway, or am I just repeating a diagram and I don't know what's going on? Okay? So your first job is to understand the material, and by working with it, diagramming it, looking up the terms, studying the pictures, drawing the pictures out, and then do I understand the variations between that by utilizing these questions or making up questions. If you did concept mapping with your 430 instructor, all right, you can take those, put the words on sticky sheets and put them on a whiteboard and say, okay, what kind of relationships can I make with these? And think of, that's another way of thinking of these questions. I can put all the enzymes in one group. I can put all the second messengers in a group. I can put all the ones that have to do with calcium activity in a group. So there's just an almost innumerous ways in which you can play, if you will, playing is still learning, with the molecules so that you're, not only you're delving into how well you understand it, but you're making your brain go over it and over it, but in different fashions, in different directions. All right? If you always take, my son still has to take his driver's test, and he told me the other day, he said, even if I take my test, I don't think I'd be able to drive. And I said, why? And he's done a learning thing, but he hasn't gotten behind the wheel. He said, I would be completely lost. I don't know how to get to different places. And I said, you've always been a passenger. Once you start driving, there's different things that you look at. You're not down on your phone playing with your phone when I'm driving. You're looking to where you're going, all right? So if you only know one direction, so if I always take him to school one way, and then while he's driving, I, blindfold him and take him somewhere else and say, now find your way home, he's going to have a hard time figuring it out because he's not going to be paying any attention to where the sun is, where other buildings are. He's not going to have a map of the freeway in his mind. And so by looking at these from different components in different directions and seeing what they have in common, which is different, then you really understand the material. Okay, you've made it, like we talked about the first day, you've made it your knowledge, not just a list of facts. Okay, the last concept that we have is to cover molecules that are lipid soluble rather than water soluble. Remember the fact that these molecules are water soluble is what prevents them from being their own messenger. From being what is the first messenger in this pathway? The hormone itself, all right? Because it cannot enter into the cell. Once it enters into the cell and binds with the receptor, it doesn't need a second messenger. So to pass across the cell membrane, it needs to be lipid soluble. So in this unit, the lipid soluble hormones that we're looking at are going to be steroids. We also had the icosanoids that we talked about yesterday, but we'll not talk about those until we get to blood, primarily. And we had thyroid hormone, and we'll talk about that again at the end of the semester. So for this unit, they're going to be steroids: testosterone, estrogens, progesterone. So here's our plasma membrane. Here's our nuclear membrane with the nuclear pores. in DNA. And our receptor can be in a couple of locations. It can be in the cytoplasm, or it can be in the karyoplasm, in the nucleus. Our hormone is lipid soluble, and so it will diffuse out of the capillary into the fluid and the loose connective tissue, cross the plasma membrane, and bind to the receptor. Or it may diffuse across the plasma membrane through the nuclear pore and bind to the receptor already inside the nucleus. Even the receptor out here is going to move into the nucleus. All right? So even if the receptor stays outside the cytoplasm, once it binds to the lipid-soluble hormone, it will still move into the nucleus. Because that's the key difference between lipid-soluble hormones and non-lipid-soluble hormones is they affect DNA. They cause, rather than causing a protein to be activated, they actually cause a new protein to be synthesized. So the receptor and its hormone interact with the DNA and result in, what do we call the process of copying the DNA into messenger RNA?
Transcription.
Transcription. So transcription occurs, and the messenger RNA moves out into the cytoplasm. Where, of course, the next step, translation occurs, and we get our new protein. That might be a structural protein, it might be another hormone, it might be an enzyme. It just depends on whatever gene was caused to be transcribed. It's going to take a short time or a long time compared to our peptide hormones? Longer. All right. So typically, steroid effects have a greater lag time between the binding of the hormone to the receptor, but they also have a more prolonged effect. And it's more difficult to turn off. Okay, we're done. The first week of the new semester is over.
15 more to go.